And the lowest plant height was showed by the result of 2,050 ppm of NaCl and 10 liters per hectare biofertilizer. And then for the number of leaves, the best productivity provided by the combination of 7,500 ppm of NaCl in controlled biofertilizer, and the lowest one is conducted by the treatment of 10 liters per hectare biofertilizer and 2,500 ppm NaCl concentration. And for the total chlorophyll content, the treatment with 0 ppm NaCl, which is the control, and 30 liters per hectare biofertilizer indicate the highest chlorophyll content compared to another treatment. And the lowest chlorophyll content was showed by the results of 0 ppm of NaCl and 0 liters per biofertilizer, or it is the control treatment. And from this research, the data that we obtained, there are plant height, number of leaves, and chlorophyll content. The treatment with 7,500 ppm and 0 liters per hectare biofertilizer indicate the highest growth and productivity compared to another treatment. In the quite high salinity concentration, the plant provides the highest growth and number of leaves without the biofertilizer given to the plant. It is because the planting medium is using manure fertilizer, which provide a directly uptake nutrient for the plant growth. 7,500 ppm is a salinity tolerance for the plant, which forced the plant to survive. The data of total chlorophyll content showed that the treatment with 0 NaCl or control treatment and 30 liters per hectare biofertilizer indicate the highest chlorophyll content compared to another treatment. The highest dose of biofertilizer provides more nutrient for the plant through its microbial activity. And this is the conclusions of this research. Biofertilizer didn't affect plant growth, but basic fertilizer does. It is affecting the plant's productivity. And the most appropriate biofertilizer dose for chlorophyll content was 30 liters per hectare. This is the bibliographical reference that we used for this presentation. I think it is enough. Thank you for the attention and good afternoon.
Good afternoon, ladies and gentlemen. Firstly, I would like to introduce myself. My name is Slamet Widianto from the Faculty of Biology, Gadjah Mada University. In this session, I would like to present my paper entitled Exploration of the Secondary Metabolite Compound of the Melon Fruit, Cucumis melon GMP, extract using LCMS. This result of the study are the initial part of the Gamma melon research project as a nutritional food, and it used as an anti-cancer. The background of the research is that the fruit are rich source of the antioxidant compound, such as a phenolic, vitamin, carotenoid, and mineral, which contribute to their chemoprotective potential. All of these substances are very useful in overcoming health problems such as the certain type of cancer, cardiovascular disease, type 2 diabetes, and also obesity. In my current research, I study about the gamma melon. Melon cultivar gamma melon parfum, or GMP, is a new melon cultivar that has been developed at the Laboratory of Genetic and Breeding, Faculty of Biology, Gadjah Mada University. Melon, or Cucumis melon, belong to the family of Cucurbitaceae with an edible fruit. Melon are a nourishing food, contain the lycopene and antioxidant. Melon possesses the ability to associate it with the reduced risk of the chronic disease. This is stimulated us to examine more deeply the benefit of the GMP melon. Cultivar Gamma melon parfum is the result on the breeding between the melon breeder and Natsumo omite and MR5 Maya Maori. Gamma melon parfum again is the phenotype characteristic stand out on the SS with a very fragrant fruit aroma like the parfum, but the taste is bitter, so it is not suitable for consumption. With this the distinctive character, it's necessary to study the bioactive compound profile contained in this KMP. So the eye of the study, the research was conducted to analyze the content secondary metabolic in the melon, commercial melon GMP using LCMS method. This is in our method. Started on the material collection and preparation, and then the extraction process. Next is the LCMS analysis, and lastly is the statistical analysis. Melon GMP was cultivated in the Agriculture Training Research Center and Development Station of Gajah Mada University, located in the Kalitirto village, Berbah Sleman, Yogyakarta. This is the process of extraction. The extraction process used the maceration method and used the ethanol as a solvent. This is the gradual method of this process. The next is the LCMS analysis. From the result of the maceration or extraction, like this pasta sample, the next collection is to process in HBLC process or the LCMS process. HBLC is the detection and the quantification method with a very high sensitivity for the various chemical compound in a particular sample using UV phase absorbent. This is referred to ANSI 2009 and also Rahman 2007. This is the result of our study. Based on the figure, you can see that the CV melon fruit in gamma melon parfum, there are the 57 secondary metabolic compounds which are grouped in the 11 extract group. These secondary metabolic compounds are ester, vitamin B, vitamin C, vitamin E, phenolic, monosaccharide, deoxysugar, flavonoid, tryptophan, cucurbitacin, and also carotenoid. The group with the highest number of the compound in the cucurbitacin compound is cucurbitacin, flavonoid, and ester, and also phenolic compound. This picture shows that from the 75 secondary metabolic compound and also 11 group in the melon fruit extract KMP, there are 5 compounds that have the lactic composition, namely chloroquinic acid, caffeic acid, catein acid, esculitin, and KM-ferol. Here are the definitive or secondary metabolic compound based on the some literature. Secondary metabolic compound of the gamma melon, such as the volatile compound like the ester and phenolic, which cause the distinctive aroma in the melon. Some function is from the metabolic compound. Many function and many use for the medicine, like as antiviral, antibacterial, and also anti-tumor, anti-oxidant, and so on. Now we can show the function of some compound from the KMP, like chloroquinic acid, caffeic acid, catein, KM-ferol, and also esculitin. And the first is chlorogenic acid, is the important of the biological active food, bilophenols, plays several important of the therapeutic roles such as the antioxidant, antibacterial, hepatoprotective, cardioprotective, anti-inflammatory, antibiotic, neuroprotective, anti-obesity, antiviral, antimicrobial, antihypertensive activity, as also as a free radical scavenger and central nervous system stimulator. Next, the caffeine acid. The caffeine acid is phenolic derivative from the various kind of plant. This function as antioxidant in various potential pharmacokinetic effect, like say anti-cancer, antibiotic, immunomodulator, also anti-inflammatory. Next, catein. Catein is a natural polyphenol compound. Its function as for various potential pharmacological effect, like antioxidative, peroxidative, also anti-inflammatory activities. Next is camphorol. Camphorol is a natural flavor, a type of flavonoid found in the variety of plant and the fruit, including the bean, tea, spinach, and broccoli. Camphorol is a potent chemopreventive agent for the cancer and protective agent for the various oxidative stress and inflammatory of the chronic related disorder. Lastly is ascoletin. Ascoletin is a derivative of coumarin with a natural lactone come from the intramuscular utilization of the cinnamic acid derivative. They have a function as antioxidant, anti-cancer, antimicrobial, and anti-inflammatory effect. From this research, I can to conclude the base of the research of the study, we was concluded that the 75 secondary metabolites were found in the chamomile perfume extract using the LCMS method. The secondary metabolic compound with the large composition in the extract is chlorogenic acid, which is in the about the 2.2%. And there are 11 group from the 70 secondary metabolic compound present in the bark of the melon fruit extract or KMP, and also in the group with the large composition is flavonoid and also phenolic acid. Secondary metabolic compound contained in the chamomile perfume has many properties for the health and beauty. So it's necessary to do further research on the properties of the melon KMP. I think it's enough. Thank you very much. Thank you for your attention.
Hello, everyone, and welcome to my presentation. Today, I'd like to present our research with the title Full-Length 16S rRNA Gene Sequencing to Detect Zoonotic Pathogens in Reptiles using Oxford Nanotomy 9. My name is Sugiono Saputra from Indonesian Institute of Sciences, or LIPI. Indonesia is the main exporter of both live reptiles and also reptile products from various species of snakes and also geckos. From wild sourced and captive breed reptiles are traded to supply the demand for pets, food, fashions, and even for traditional medicine. From 2000 until 2015, more than 1 million live reptiles from Indonesia had been transferred to the USA. And the traffic for this animal trade is quite high, not only to the USA, but also to Europe and to several Asian countries. But unfortunately, the potential pathogens from these Indonesian reptiles remains unexplored. Even we know that the emergence of novel zoonotic pathogen is one of the greatest challenges to the global health security. So in this study, we aim to detect pathogens from reptiles using next generation sequencing approach, which is allow us to detect and to identify the difficult to culture or unculturable bacteria, which can be complement with a culture dependent method. For the last decade, NGS instruments are less expensive, faster, and even smaller, and have been widely used in several applications, including for pathogen detections. And with this device, Minion sequencer, we will obtain long read and also better resolution. It can be useful to discriminate between members of bacteria. So this is our experimental workflow. We obtained a snake and also a gecko fecal sample from slaughterhouses in Cirebon, West Java, and then performed DNA extraction with a Presto stool DNA extraction kit from Gen8, Taiwan. And in-house 16S PCR was performed using 27 forward and also 1492 reverse primer with approximately resulting 1.5 kb. The DNA library preparation was conducted using ligation sequencing kit LSK109 from Oxford Nanopore Technologies, and the sequencing itself performed in MinION Mach1B device. The best calling performed in MinNo software, and also analysis of the base call reads was performed using the 16S analysis in Epitomy. In snake sample, we observe 25 phyla with dominated by Fusobacteria with more than 40%, followed by Firmicutes and Proteobacteria. At the genus level, more than 130 genus were detected, dominated by Clostridium, Plasmonas, and also Bacteroides. We also observed that the proportion of Enterobacteriaceae was 0.5 of total reads, and some notable pathogens were observed, including Salmonella, Shigella, Vibrio, and also Yersinia. The different result was observed in gecko samples with more diverse of phyla and also genus at the genus level. But in gecko samples, the dominant phyla and also dominant genus were significantly different with snake samples. For example, at the genus level, more than 660 genus were detected, dominate with Lachnoclostridium, Bacteroides, and Akkermansia. And also the proportion of the Enterobacteriaceae was also very low, with 10 times less than compared with snake samples with 0.04% of total reads. Even we also find that Salmonella, Vibrio, Shigella, and also observed in these samples. But for Leptospira, we did not observe in snake sample, but only observe in gecko samples. To conclude, at least 3 points. First one is the number of reads obtained from MinION sequencer classified were higher than the sequencing random fragments from whole genome due to the full length of 16S genes, approximately 1.5 kb. And also several notable zoonotic pathogens such as Salmonella, Listeria, Shigella, and Vibrio were detected in both samples, but Yersinia was observed only in snake samples, while Leptospira only observed in gecko samples. These methods, full-length 16S sequencing, offer comprehensive detection of multiple pathogens and as a complementary to detection by conventional PCR and culture. And I think that's all from me, and happy to answer if there's any questions. Thank you.
Okay, thank you for all the presentation. That was all a splendid presentation from our first three presenters of this subsession. So I would like to ask the three of the presenters to turn on their camera again so that we can interactive discussion. Okay, wait, let's see the chat box. Okay, we have some questions here. So we will start with the first presenter. Just share my screen. Okay, so this is the first question for the first presenter. So the question is, it is mentioned that the biofertilizer doesn't affect plants' growth, but does affect chlorophyll content, right? So is it possible to combine both biofertilizer with those content inside basic fertilizer, which provide plants growth, so that we can make the plants grow fast and have a good chlorophyll content? Or is it an impossible aspect to reach due to some like special reaction? Miss Nadifa, you may answer the question.
Okay. Beforehand, I would like to thank RTI grant from Universitas Gadjah Mada for making this research possible to do, and thank you so much for the question. Yeah. Can I repeat the question? So it is mentioned that biofertilizer doesn't affect plants' growth, but does affects chlorophyll content. Is it possible to combine both biofertilizer with those content inside basic fertilizer, which provide plants growth, so that we can make plants grow fast and have good chlorophyll content? So yes, biofertilizer can be combined maybe with another basic fertilizer like NPK or such thing for activating the microbia beside the biofertilizer, like that.
Okay, so it's possible to reach the question
because the biofertilizer needs a basic fertilizer to become an active fertilizer.
Okay. Okay, thank you, Ms. Nadifa. We will wait for other question. And okay, moving on to the second presenter. Okay, so we have a question here. So the question is, why you choose LCMS, not GCMS, and what are the differences between LCMS and GCMS to analysis?
I'm sorry, I can go here any. I don't know. Yeah, we're using LCMS because it's this in our laboratories. We used to analyze some biocompound using these methods. So just easy to use and maybe to more easy to analysis this using this method. Hello?
Okay.
Yeah.
Okay, thank you.
Can I also ask some questions to Mr. Slamet as well?
Yes, Dr. Christiana.
I'm just wondering, so it's based on its component, the melon or the GMP contains lots of goodness. So when you did the analysis, did you do as a timeline? I mean, like when it's young until it's ripe? Because I'm curious whether one compound may elevate or degrade during the certain time. Also during the cultivation, is there any particular condition that may support the components of the GMP? I mean, the goodness of there. And what will be your next approach to promote the advantage of the GMP consumption?
Yeah, I analyzed many some state of the fruit, maybe in the early and or also in the fruit in the final harvest. So I analyze and compare, and this is different content. It's very different. So some compound is maybe in the middle fruit, very good for maybe I just try some effect in anti-cancer. It's very good, but in anti-diabetic is not good because I don't know. Maybe I will going to research more about this compound. No, I still purified this compound, and also I try in vivo and in vitro for anti-cancer.
It's very interesting that that one component is going up and down at a certain time. Like you don't know yet what is influence that, or is it because of the fertilizer? Is it because of the season maybe or temperature?
Yeah. Maybe next I will to continue my research about many factors for impact in level of biocompound.
Yeah, we're looking forward to hear that from you. Thanks.
Thanks, Dr. Slam. Thanks, Mr. Slamet, for the explanation. Okay, so we will move on to the third presenter, Mr. Sugiono. So we have a quick question here. I'm going to share. So this is the question for Mr. Sugiono. Did you compare the rates relative to the host genome, and how are the quality and length of important pathogens rate that could be retrieved by Minion when compared by other NGS? Thank you.
Thank you for the questions. So we conducted the 16x RNA sequencing from fecal samples, and we didn't compare with the host genome. So it's completely from the fecal samples. And then for the second question, with the full length, we obtained the amplicon with the length about 1,500 base pairs. And then during the sequencing, we obtained a relatively diverse sequence. But the cutoff that we obtained for the next analysis is between 1.4 to 1.7 kb base pairs. Yeah.
Okay. Thank you for the explanation, Mr. Sugiono. I think that was all for the question. I have one more question for Dr. Slamet, I think. Yeah. I think we still have time, yes, for Mr. Slamet. So the question is, how much is the chlorogenic compound in GMP, which is more profitable to take this compound from GMP melon with high plant maintenance costs, or from Valeria macrocarpa or bitter melon?
Yeah, maybe the question is more. I can read this question. Yeah, okay. In melon, chlorogenic about 7.2%. So maybe this very high, they depend on another plant. But in maybe in what's mean to the high plant maintenance cost. Related to textile compound. Oh, sorry, I can read, but the compound of chlorogate is about maybe about 7.2%. I think it's, this is the height depend on compare in another melon.
Okay. Yeah. Okay, Doc. Thank you, Mr. Slamet, for the explanation. I think that was all like a good start for our first subsession. I think we can move on to the next subsession. Dr. Christiana, you may take over.
All right, thank you, Andre. So for the next subsession, we'll have another three presenters. So we'll have Herjuno Ari Nugroho, and Didit Tulus Subakti and Fitrin Ekawasti. So Herjuno will present the development and optimization of primer sets used to study the relative expression of androgen receptor gene in Turkey, and then followed by recombinant protein of SEC1 and native protein for toxoplasmosis detection, and it will end with this restriction fragment length polymorphism or RFLP analysis genes of Toxoplasma gondii using enzyme DDE1. All right. To the co-host, you may start the video. Thank you.
Good afternoon, everyone. Thank you for International Conference of Advanced Veterinary Science and Technologies for Sustainable Development, or ICAVESS 2021, for the opportunity given to me. My name is Herjuno Ari. I'm with Research Center for Biology, Indonesian Institute of Sciences, LIPI. I want to make a short presentation about my study entitled The Development and Optimization of Primer Sets Used to Study the Relative Expression of Androgen Receptor Gene in Turkey, Meleagris galopevo. The research was conducted in a collaboration between LIPI and Faculty of Veterinary Medicine, Universitas Gadjah Mada, represented by Prof. Rini Widayanti, Dr. Triwahyu Pangestiningsih, and Mrs. Ali Supriyani. Let me start with some background and general information about our research. The turkey is initially native to the North America and widely captivated in several countries mainly as protein source. In the captivity, reproduction of the birds plays important roles. The expression of androgen receptor or AR genes mRNA can be used as information about genes regulation that related to reproduction performance. IR itself is expressed in many organs, for instance, in the testes to support spermatogenesis regulation. The objection of this research were to develop and optimize new primer sets for relative expression study on Meleagris galopevo IR gene mRNA. The research was started by submitting ethical approval to Faculty of Veterinary Medicine Ethical Clearance Committee. The research was approved by protocol number as follows. The primer were designed using primer 3+ and in silico tests for specificity was conducted using primer plus. The design primer as follows. The primer that targeting RR gene was called AR. Meanwhile, the primer for beta-actin genes for internal control was called PAK primer, and the product size as follows. Samples were collected from fresh testicle from 2 jigs and then processed for cDNA synthesis. Primer specificity test was conducted by PCR amplicon sequencing followed by nucleotide plus. Primer efficiency were tested in qPCR using 3 levels of sizes of serial dilution. According to in silico test, the primer were annealed to the corresponding target. PCR was conducted to amplify the target. The amplicon then conducted electrophoresis and showed that IR primer produced around 250 something base pair and PIK primer amplify around 160 something base pair amplicons. We conducted single sequencing on this amplicon and conducted nucleotide class. The results show about 98 to 100% similarity with Malacryskalopavugen. Primer concentration optimization were conducted on 3-fold of serial dilution on both primer. According to Kwang et al., ideal CT value should be around 20 to 30. We found that 0.1 micromolar primer produce ideal CT value for both primer. For each primer, we conducted primer efficiency tests by performing PCR on 3-fold of serial dilution. We calculated efficiency using standard curve. According to Kwang et al., efficiency for primer should be around 93 to 103%. Meanwhile, our primer efficiency is 98.03%. We also conducted efficiency calculation on PAK primer using the same procedure. As Kwang et al. Stated, efficiency should be around 93 to 103%, and our primer efficiency is 98.01%. From our research, we conclude that the primer were able to amplify their respective targets. The specificity was proved by several tests, and both primer passed minimum primer efficiency parameters according to Kwang et al. But tests on larger number and multiplication should be performed. We thank to RTA program 2018 from Universitas Gadjah Mada for funding this research. That's all my presentation. Thank you for your attention.
Assalamualaikum warahmatullahi wabarakatuh. Hello everyone, thank you for having me. Let me introduce myself to you. My name is Dewi Marbawati. I am the second author of this paper, and I'm going to present our article titled Performance of Enzyme Immunoassay Using Native Protein versus Recombinant SAG1 Protein for Toxoplasmosis Detection, for the first International Conference of Advanced Veterinary Science and Technologies for Sustainable Development. And this research based on our collaboration research. So let's get started for. Toxoplasmosis is transboundary disease infect animal and human without any national restriction. Toxoplasmosis in Indonesia was relatively high. Banjarnegara Health Research and Development Center took the initiative to develop ELISA enzyme immunosorbent assay-based toxoplasmosis diagnostic kit. We choose the ELISA because most suitable and relatively easy technique and widely applied for mass diagnosis, while the commercial kit is relatively expensive for health services in Indonesia. This study will evaluate and compare the performance of the ELISA test using native and recombinant agents to detect anti-toxoplasmotic antibody IgG. The native antigen we use is soluble protein, that produce higher accuracy, while the recombinant protein we use is SAG1 that more sensitive than other recombinant protein. For methodology, for serum sample, a total of 207 human serum sample from Banjarnegara and Banyumas district of Central Java Province, Indonesia was used in this study. And to determine the true positive and true negative serum, we used two commercial ELISA kits. All serum samples were used for Toxoplasma IgG using two different commercial kits. This assay to establish true positive and true negative serum. Validation is carried out in two stages. First, all sera test by RSAG1 ELISA and native protein ELISA analyzed using the ROC curve. Based on ROC curve are known predictive values of sensitivity, specificity, and cutoff. For the second, the absorbance value is converted to a score. When the score of absorbance value of sample per absorbance value of negative control more than 1 in a value, we designated it as positive sample and the other was negative sample. Samples that have been declared positive, negative, false positive, and false negative are tabulated and analyzed with diagnostic tests using MedCap software. For antigen preparation, Toxoplasma gondii, we ground Toxoplasma gondii in ferrocile and through several stages to get the native protein. And for the ELISA protocol, we carry out the usual ELISA step until we read the ELISA result into ELISA reader. And for data analysis, we use receiver operating characteristic curve, continued with diagnostic test MedCalc and analysis interreliability agristat. Diagnostic performance of RSAG1 ELISA using the ROC curve result that the sensitivity and specificity of RSAG1 ELISA was 84.6% and 90.3%. These results are almost the same as other studies, but when the recombinant protein are combined, for example, with 3 recombinant protein, the result of sensitivity was increased. Several studies reported high test performance, the use of recombinant protein, a single for ELISA, but there are some notes of criticism. Notes of criticism is serum sample used for validation were predominantly negative sera with low antibody levels and positive sera with high antibody levels. The diagnostic performance of NP ELISA using the ROC curve saw that the sensitivity and specificity of NP ELISA was 92.8% and 97.4%. Research using NP ELISA from peritoneal mice saw that the sensitivity and specificity value lower than NP protein from cell culture. Postbin compared the sensitivity and specificity of ELISA, native protein from cell culture, and from peritoneal mice, and the result was significantly different. Researchers in 2012 proved that one of the critical points in the problem of native protein quality in the purification technique and the consistency of native protein profile on SDS page. Same with Vera done in her research. This proved that the use of Toxoplasma gondii from peritoneal mice, which is purified with adequate technique, will produce consistent protein quality and profile, one of the important factors to produce high ELISA sensitivity and specificity. Comparative performance between RSAG1 ELISA versus NP ELISA. Of course, NP ELISA has a better diagnostic test performance than RSAG1 ELISA. However, statistically, RSAG1 ELISA is still acceptable and feasible to apply. And in general, the test result between RSAG1 ELISA versus NP ELISA and also NP ELISA was also categorized as having good fit. This categorized refers to Altman research. For the conclusion, first, the use of coated recombinant protein-based antigen would reduce the diagnostic sensitivity of ELISA. Second, native protein for ELISA toxoplasmosis is more recommended than recombinant protein. And for the last, the performance of the toxoplasmosis ELISA using combination of several recombinant protein will get more consistent result and equivalent to native protein. So this is the end of our presentation. Thank you very much for the attention. See ya. Wassalamualaikum warahmatullahi wabarakatuh.
Assalamualaikum warahmatullahi wabarakatuh. Thank you very much for this opportunity. Let me introduce myself is Fitrina Ekawasi and our team are Umi Caheningsi, NLP Indi Darmayanti, Siti Sadia, Didi Tulus Bekti, Zul Azmi, and Muhammad Ibrahim Desem. We are from Indonesia Research Center for Veterinary Science, or BBLITVET, and IPB University. In this opportunity, I would like to present our research about restriction fragment length polymorphism, or RFLP analysis, genes of virulent strains isolate of Toxoplasma gondii using enzyme DDE1. Introduction. Toxoplasma gondii is one of the most common human zoonoses. It can infect a wide range of warm-blooded animals, including humans. For the global status of Toxoplasma gondii in human, about 43 until 88%, and then in animals about 6 until 70%. Toxoplasma gondii also known to cause economic losses in the livestock industry due to abortion, premature, and stillbirth. Infection with Toxoplasma gondii in human is usually asymptomatic. However, the infection may cause abortion in pregnant women or occasionally toxoplasmic encephalitis. Toxoplasma gondii has been considered a single species in the genus Toxoplasma. Clinical symptoms of Toxoplasma gondii infection are non-specific and unreliable for diagnosis. The detection of virulent strains of Toxoplasma gondii infection by molecular methods high sensitivity and specificity, but by PCR-RFLP fragment length polymorphism is popular technique for genotyping. PCR-RFLP is a genetic polymorphism of different Toxoplasma gondii strains was studied by PCR-RFLP using the following markers with enzyme DDE1. PCR-RFLP using the DDE1 to characterize the differences between virulent and virulent strains. DNA target regions used in this study that are important for Toxoplasma gondii include genes B1, SAG1, P30, BAG1, ROP1, GRA1, and GRA7. They are frequently being used for genotyping of the isolate worldwide because specific and the most sensitive for Toxoplasma gondii detection. The purpose of our research to identify the sets of PCR-RFLP markers that are easy to use and can provide higher resolution for Toxoplasma gondii genotyping, to identify the virulent strands isolates of Toxoplasma gondii by PCR-RFLP using 10 markers with the GE1 enzyme restriction. The methodology, DNA isolation, Toxoplasma gondii tachyzoid from RH virulent strands was derived from culture cell. The DNA template was extracted using the purine genomic DNA mini-kit from in vitro gen, and then you purity the concentration of DNA extract for DNA quantification using the thermoscientific nanodrop. And then for gene analysis, we did a PCR and then DNA sequencing by Bioner sequencing, and then RFLP. This is in brief for the methodology. We use tachyzoid of Toxoplasma gondii and then DNA extraction, and then we perform the amplification and then the PCR product sequence and analysis by CLC sequence viewer version 8 software. After that, we choose the restriction enzyme. We use GGA1 because GGA1 is common restriction enzyme used in laboratory. And then the procedure enzyme treatment was conducted from Promega company. After that, we visualization the restriction pattern using a UV transluminator, then compare the experiment from the electrophoresis gel, then in silico analysis. Result and discussion. Although the genus Toxoplasma consists of one species, Toxoplasma gondii, but molecular biology tools have provided the evidence of 3 distinct clonal lineages of Toxoplasma gondii, biotype 1, 2, and 3. The intersection by restriction enzyme GE1 reaction by PCR-RFLP can be predicted by in silico analysis using the gene sequences from PCR product, or the real reaction PCR-RFLP can be seen from the gel electrophoresis visualization. This is in silico restriction site analysis of gene sequences digest using enzyme GE1. In silico is one performed by a computer simulation to predict the expected DNA segments. In this study, we use CLC sequence viewer version 8 software. And then from the table, we can see 6 genes have restriction sites B11, B13, BAG1, ROP1, GRA1, and GRA7. And then we can see from the line in pink color, the pink color is a number of cut sites less than or equal to. There are B11, BAG1, and GRA7. And then in the line, the yellow color, we can see the number cut. The number of cut is more than 2 are B13, ROP1, and GRA1. And then we compare to the PCR-RFLP analysis real from the electrophoresis gel visualization from the figure. Based on the result of the electrophoresis, we can see all DNA sample from Toxoplasma gondii virulent of the electrophoresis isolate were amplification. And then PCR-RFLP with restriction enzyme GE1 performed among 10 genetic markers of Toxoplasma gondii. Only 3 genes could produce intersection of fragment pattern. This is different from in silico analysis, which shows 6 genes that produce restriction sites. Besides that, from the figure, we can see the other analysis from the B1 gene. We use 3 kinds of B1 gene, B11, B12, and B13. Based on the result of PCR, 3 primers of B1 gene successfully amplification well, but after being digested by the enzyme, not all B1 genes saw intersection fragment, and compared in the silico analysis, the restriction enzyme cut incomplete or didn't cut the DNA in a real experiment. The B1 gene in PCR gene successfully amplified well, but in RFLP only 1 gene saw intersection fragment. The origin of the same target gene, but a different arrangement of the primer genes that create different results. Identify the capacity of the pair of primers necessary to evaluate DNA sequence and needed an optimization method. For analysis of incomplete or no digestion gene, based on this study, not all genes that were successfully digested using restriction enzyme DDE1 by PCR-RFLP. Incomplete digestion is the most occurring reaction in PCR-RFLP. Many factors can affect the reaction, such as size, the DNA fragment, and optimization the method. The amplified product is restricted with the DDE1 enzyme, which recognize the cutting site. Conclusion: the specificity and sensitivity of values of a variety of PCR-RFLP methods for virulent strains of Toxoplasma gondii diagnosis are depend on several factors, including the primer gene target and restriction enzyme. 3 pairs of primer B13, ROP1, and GRA1 with GA1 have proven useful, and this method can be used for rapid diagnosis acute toxoplasmosis, while the other genes and restriction enzyme must be optimization. PCR-RFLP is specific, sensitive, easy to perform, and ready to be adopted for diagnosis and genotyping study of Toxoplasma gondii. Thank you very much for your attention. Wassalamualaikum warahmatullahi wabarakatuh.
I'm mute myself. All right, I'll call for some questions. Okay. Okay, there's a question for Mr. Herjuno.
Okay.
Yep. It's saying that based on your result, when we should do the test for expression of AR or androgen receptor in Turkey, do you have any range age of Turkey to test the AR gene?
Can you hear me? Sorry.
Yes.
Oh, yes. Thank you very much for the question. I'm trying to answer the question. About the primer is actually according to a previous study, the AR gene transcript or the AR gene mRNA is actually start to emerge for the early age around a few weeks after hatchling of the turkey chicks.
Okay. All right. So if we do the test on the older turkey or mature turkey, it won't work. Is that what you're going to say?
Yeah, because actually AR expression is a dynamic process in the turkey. It will rise in the early age and will decrease in the older age. It actually will work unless there are no expression for AR anymore in the older turkey, probably. But it's actually already being studied by previous study by several researchers in China, and they actually use different primer, and but they designed the primer from chicken gene reference. So we try different primer, a new primer from turkey reference gene.
Okay. So you're hoping to get more CT values in what younger turkey compared to older? Is that what you're saying?
Yeah, hopefully.
Okay, so you haven't really tested. How old was the turkey that you used before in your experiment?
Actually, we already conducted several tests in younger turkey and older turkey, but we only conducted in 1 year old turkey and over 1 year old turkey. But we not conducting in the younger turkey yet.
Okay, thank you. So So you really think that the diversity of the population might influence the reproduction performance of the turkey in Indonesia? Is that what you're implying to?
It's really hard to answer since we don't we still we're still running the study, but hopefully it will show the result like that. So like that.
All right, thank you, Mr. Herjuno.
You're welcome.
Okay, so we have a next question for Miss Fitrina. Am I right of saying your name? So what would you recommend to further study on detecting other genes that you mentioned need to be optimized? So I think it's talking about the B1 gene, I think. Still unmute.
Okay, sorry. Can you hear me now?
Yes.
Yes, I recommended the gene for B1 to optimization first to do the RFLP, because from to do the RFLP, we can must to do optimization the PCR, single PCR or nested PCR from to do a PCR-RFLP. Because if we see from the in silico analysis and then we compare then the electrophoresis gel, it is the different result. It is according to the optimization for the squam gel and then the visualization for electrophoresis gel. I think that's all.
But you did sequencing, right?
Yes, I did the sequencing.
And you see the cutting area, so it should cut. So the DDA one should cut somewhere in the B1.
Yes, and in D1 D1 gene, so the restriction site from the D1, but we use 3 kind of B1 gene. But in electrophoresis gel, we just produce one of the B1 gene. But in silico analysis, we get the 2 gene restriction site of B1.
So what's the difference between those 3 samples that they use? Is it the biotype 1, 2, 3? Is that what you're saying?
Yes.
So
Yes.
Something to do with so probably the inefficient cutting for the DDE is something to do with its pathogenicity, do you think?
Yes, we use the biotype 1 of toxoplasmosis, Toxoplasma gondii is the pathogenetic, and then we use 3 kind of B1. 3 kind of B1 is the different construct of the sequence of primer. So we get the different result from there.
All right. Okay. Thank you so much. That's very interesting work.
You.
Okay, so do we have another one? Okay, so probably a question for Ibu Dewi Marbawati. I'm just wondering, how did you do with the recombinant? You haven't explained how did you construct the recombinant? Or is it ready-made, and then you just use the protein? Unmute, please. Ibu Dewi.
Can you hear me? Sorry?
Yes.
I'm sorry, I use my mask because I'm in public area.
It's fine. Yes. Me too. Certainly fine.
Repeat your question, please.
Yep. I haven't seen the way how you construct your recombinant. I was wondering, how did you do that?
For the recombinant protein used in this research, we buy it in we buy it. We not produced by yeah.
Okay, so you just produce your native protein by yourself in the mouse, is that right? But you didn't make your own recombinant. So is it in terms of as a protein, like is it expressed and then it's purified?
Yes.
Okay. Have you ever thought of the unsuccessful or is it the less binding that creates by, if you compare both between the native protein and the recombinant, is it because the antigen might have the conformational epitope?
We just by ELISA assay, and we repeat the assay in several parts, and for recombinant, we do the combine the recombinant protein too, but the data not found in this research, but we prove that the combine of recombinant protein will have a good result in the performance of recombinant protein.
Okay, so so let me summarize. So it's combination between the native and the recombinant, what you're saying?
Oh no, for only for recombinant protein.
Okay, all right. Okay, so yeah, I'm just wondering because it's
yeah.
Sorry,
I am the second of this author, and the principal investigator of this research was on the board. Mr. D. Maybe he will clarify my answer to explain more clearly for this research. Maybe.
All right, Bapak Didi, would you like to clarify or? Yeah. Yeah. Would you like to clarify, Bapak Didi? You want to add something?
Okay, thank you. I'm sorry. Could you inform to us what the question is?
It's okay. We were talking about the antigen that you're using on your ELISA. I assume that you were trying to make your in-house ELISA, and then you're comparing between native protein compared to recombinant, which is store-bought. Like it's you're not producing your own recombinant. And then it turns out that the native protein turns out giving better binding rather than the recombinant. So my question was I'm just wondering whether it's something to do with the conformation of the antigen itself, like the epitope itself.
No. Okay. I think that probably recombinant protein only one protein. Whatever about the conformations. In ELISA, we make based on, I'm sorry, the noise in my voice, okay?
Yep.
It's clear?
Yep. Yeah, a little bit small, but it's fine. But I can still hear you.
But the ELISA more influenced by the content of protein. Multi-protein is good for to make high sensitivity and high specificity. If we use recombinant protein, that's good for specificity, but sensitivity usually low because we only use one antigen. when we use native protein, we get more kind of protein. So we can optimize with such immunoblotting to optimize how many the dilution of samples, conjugate, and any other. That native protein is good or suitable if we want to get of high sensitivity and high specificity, not influenced by conformational, but more kind of protein, not single protein. We can do to adjust.
All right. Yeah.
Okay.
Yeah, I certainly agree with you that native protein is the best source, but thinking of the production, the mass production, and you're thinking of making your own in-house ELISA, probably recombinant would be much easier to produce, maybe.
For toxoplasma, it's not. We produce by cell culture, so we can get the whole protein, pure protein, only in 1 day, maximum in 2 days. So the protein, when we can prepare with good treatment, it's enough for years, or 1 microgram. 1 microgram is enough for 6 kit.
Sorry, for
6 split.
60 or 6?
6 split.
So 100 samples.
100 samples, yes.
96 samples minus control 80 samples.
When we compare with recombinant, the cost is still high.
Because you bought it, maybe.
No. We have already make the recombinant with Dr. Wayan in 2000 until 2007.
Okay, all right. Is it in
still the cost is still high and not worth it with sensitivity.
Okay. All right. Thank you so much. I'm not going to.
You're welcome.
All right. We should move to the next session. I'll hand it over to my colleague Andre. Time is yours.
Okay. Thank you, Dr. Christiana. So I would like to start our third session. I think it's the discussion is getting warmer and warmer. I hope we can have a good discussion for the last 3 subsessions. So I would like to ask Mr. Desem, Ms. Gayana, and Mr. Subekti to ready to turn on your camera when it's needed. So for the third subsession, we will have 3 presentations. For the first presentation, it's about the effectiveness of API 2.1 kit for Passerella multocida identification by Mr. Muhammad Ibrahim Desem, followed by a presentation about farmers' empowerment through managing organic waste as fertilizer in Camplong Second Village, Kupang Regency by Cynthia Dewi Gaina. And the third presentation is about evaluation of the utilization of GRA7 and ANDSAG1 genes to predict virulence of Toxoplasma gondii by Mr. Didik Tulus Subekti. Okay, without further ado, I would like to ask co-host to play the video presentation. Okay, enjoy.
Assalamualaikum warahmatullahi wabarakatuh. Thank you to the committee of the first International Conference of Advanced Veterinary Science and Technologies for Sustainable Development for giving us the opportunity to make this presentation. First of all, allow me to introduce myself. My name is Muhammad Desem, and I will present our paper entitled The Effectiveness of API 20.1 kit for Pastorella multocida identification. This paper was co-authored with my colleagues at Indonesian Research Center for Veterinary Science, Dr. Didik T. Subakti and Dr. Susan M. Noor. As an introduction, I will tell you about Pastorella multocida characteristics to refresh our memory about it. We know that it is a gram-negative bacteria. Its shape is rod and often look like Coccobacilli bacteria. This bacteria is well known as a causative agent for septicemia epizootica in cattle and buffalo. We know that this disease is still a major problem in Indonesia, and it is still enlisted as strategic transmissible animal disease or PHMS. Pastorella multocida is non-motile bacteria, and we can see that some of its important biochemical characteristics for identification are its type of hemolysis, which is gamma hemolysis, oxidase positive, producing indole, and lack of growth on McConkie agar. This is a problem we have encountered with bacterial identification in general, and this is the logical framework for the research we did. We have seen that the majority of bacterial identification methods use conventional methods. This method generally consists of observing the phenotypic characteristics and biochemical testing. This method is not efficient because it takes a lot of time. Therefore, testing that can identify bacteria quickly and accurately is a necessity at this time. In this case, API 20NE is here to meet these needs. API is said to have high accuracy, but some reports say that it has potential for misinterpretation in Burkholderia and Enterobacteriaceae. So it is raised the question, is it effective for pastoral identification or not? In this study, we used 10 bacterial isolates from IRCVS collection. The arrangement of these bacteria was performed in brain-heart infusion broth medium and then cultured in blood agar medium. Bacterial confirmation was carried out using two methods. The first one is by Cohen's algorithm and the second is by API 20NE. The tests that included the Cohen's algorithm are gram stain, catalase, oxidase, indole production, carbohydrate utilization, growth on McConkie agar, and motility. This is the results of our research, and as we can see that the characteristics that observed in all isolates were gram-negative, and its shape is rod or Coccobacilli as we seen on the figure B, and its type hemolysis is type gamma hemolysis or non-hemolysis as we can see on figure A. And the other characteristic is oxidase and catalase positive, producing indole, non-motile bacteria, and lack of growth on McConkie agar except for two isolates, as we can see on the figure C. And on the AP20NE results, 8 isolates correctly identified as Pastorella multocida and gave numerical profile 3000004, and with percentage identification is 96%. Meanwhile, one isolate misidentified as Vibrio parahemolyticus and gave numerical profile of 7067746 with percentage identification of 98.9%. And another one isolate is misidentified as Aeromonas hydrophila and gave numerical profile 7746 with percentage ID 87%. From this result, we can say that the effectiveness of API 20NE for Pastorella multocida identification is 80%. Two isolates that misidentified by API were confirmed as Pastorella multocida by traditional assays. This test refers to Berge's Manual of Determinative Bacteriology, and it uses various carbohydrates fermentation as an indicator. From the results, we can also know that all the bacteria used are non-motile bacteria. Meanwhile, we can see that Vibrio and Aeromonas are motile bacteria. This is one of the distinguishing characteristics that can be used to distinguish Pastorella from Vibrio and Aeromonas. This characteristic is not accommodated by AP20NA. And we can see that pink colonies on McConaughey agar is show the characteristic of the bacteria is lactose fermenting bacteria. This is uncommon characteristic on Pastorella multocida, but in very rare cases, lactose fermenting Pastorella multocida has been reported before by Talbot and Smith in 1960, by Holmes and Brandon in 1965, and then by Hiddleston and Westman in 1975. We can see that API identification system mostly rely on the information about bacterial ability to utilize carbohydrates. So if there are two species with similar carbohydrates utilization characteristics, the API system will detect them as one species. Apart from the motility characteristics problem previously mentioned, the API system also cannot accommodate hemolysis as distinguishing characteristic. In general, Aeromonas bacteria are beta hemolytic type, whereas Pastorella multocida is gamma hemolysis type, or in other words, non-hemolytic type. And this was also reported by previous study that Pastorella multocida also misidentified as Aeromonas salmonicida and Vibrio halisae by Lizarazo et al. In 2008. So as a conclusion from the results in this study and previous reports, the API 20NE kit is not 100% accurate and effective for Pastorella multocida identification. Therefore, it is recommended to use it as a complement to traditional identification systems to give more reliable results. That's our presentation. Thank you for your attention. Wassalamualaikum warahmatullahi wabarakatuh.
Thank you, ladies and gentlemen. Thank you for opportunity and honor for us to presenting our study. The title of our study is Toxoplasma gondii virulence prediction using hierarchical cluster analysis based on SAC1, GRA7, and ROB18 coding sequences. Toxoplasma gondii is one of the most common zoonotic diseases and reported to infect nearly a third of the world's population. Toxoplasma gondii is also known to be one of the causes of miscarriage in human and animal, especially goat and sheep. The successful isolation of various Toxoplasma gondii strains around the world has led to the knowledge of genetic variation existence in many isolates. This variation can be seen from 2 points of view, namely virulence and clonal population. Toxoplasma gondii virulence determination is based on its pathogenicity in mice, while a clonal population based on genetic analysis by PCR-RFLP, which is associated with its virulence. Toxoplasma gondii virulence classification by mouse bioassay group it into 3 groups. The first group is virulent strain. This group 100% lethal to the rodent at infective dose 1 tachyzoid per mice in a week. Induce hypersecretion of pro-inflammatory cytokines or cytokine storm in rodent, and also difficult to form tissues on rodent. The second group is a virulent strain with LD50 more than 10 to the power of 3. This group is characterized with 50% lethal to the rodent at infective dose 1000 tachyzoid per mice in a week. They also induce intermediate secretion of pro-inflammatory cytokines in rodent and easily to form tissues on rodents. The third group is a virulent with LD50 more than 10 to the power of 5, which character 50% lethal to the rodent at infective dose 100,000 tachyzoid per mice in a week. They grow well on immunosuppressed rodent and usually easily eliminated by immune system of rodent or self-limiting disease. And the other side, Toxoplasma gondii virulence classification using PCR-RFLP require several multi-locus genetic marker and many different restriction enzymes. However, they resulted in consistent and inconclusive electrophoretic patterns are frequently obtained. Based on PCR-RFLP, there are two groups, namely archetypal lineage and non-archetypal lineage. Archetypal lineage is divided into three groups. Type 1, which is correspond to virulent strain, while type 2 and 3 correspond to a virulent strain with LD50 more than 10 to the power of 3 and 5 respectively. Unfortunately, there are circumstances where tachyzoid isolation fails to be achieved or cannot grow for any reasons. So, a mouse bioassay cannot be implemented and PCR-RFLP is also difficult to realize. In this situation, we are required to looking for another alternative method, namely hierarchical cluster analysis, which is based on a complete coding sequence of SOC1, RA7, and ROP18 genes. How to perform it? Briefly, DNA or mRNA is extracted from live or dead tachyzoid. And then DNA or mRNA is amplified and sequenced. Finally, coding sequence was then aligned and analyzed by hierarchical cluster analysis. This is the results of analysis. Clustering of Toxoplasma gondii isolates is constructed by UPGMA method on SAC1 CDS. This group, the red one, is virulent strain or type 1. And the green color is group of avirulent strain, which is type 2 or type 3. In figure 2, clustering of Toxoplasma gondii isolate is constructed by UPGMA method based on RA7 CDS. They able to differentiate into three groups. The green groups is a virulent strain or type 3. The purple group is a virulent strain or type 2. And the red one is virulent strain or type 1. This is clustering of Toxoplasma gondii isolates based on ROP18 CDS. They also differentiate into three groups. The purple one is a virulent strain or type 2. The green group is a virulent strain or type 3. And the red group is virulent strain or type 1. SOC1 can classify Toxoplasma gondii into virulent and avirulent as well. Unfortunately, the avirulent group could not be separated into type 2 and type 3. On the other hand, RA7 also successful to classify Toxoplasma gondii into virulent and avirulent as well. RA7 is able to differentiate a virulent type 2, virulent strain type 3. And RA7 were good at differentiating between virulent and avirulent strain in archetypal lineage. But ROP18 is actually quite good, but has a minor misplacement. Thank you for your attention and have a nice day.
including fellow. Good afternoon and greetings to all of participants, including fellow veterinarians. Now, I'm going to present our topic entitled, let me share first. Entitled Farmers Empowerment Through Managing Organic Waste as Fertilizer in Camplong to Village, Kupang Regency, by me, Cynthia Dewi Kaina, and other colleagues from Faculty of Veterinary Medicine in Universitas Nusa Cendana, Kupang, Indonesia, and also Faculty of Animal Science in the same university. The background of this research was actually based on the low productivity of livestock waste in Camplong to Village. It is caused by a lack of knowledge and skill owned by group members related to integrated livestock farming management. In the integrated livestock farming management here, including unuse of livestock waste and animal health management issue. Based on this reason, a collaboration between FKH Undana and Camplong to Villages has been established since 2017 to assist farmers' community to enhance local farmers' knowledge and skill in livestock management, such as animal health management and waste management. This is according to Kupang Region's Regulation number 4, 2015, regarding village fund priority should be in line with the achievement of the village development plan and village government work plan. Village farmers in Camplong too, as the main actors of income sources for their families, have been dealing with uncertainty and risk during this day as their livestock-dependent societies. Local farmer has enough knowledge and skill in terms of common livestock management practices, but their knowledge in the use of organic manners that they got it so much from animal and green manners are limited. So one of the activities that we transferred to this community was the making of organic fertilizers from co-manure called bokasi. Indeed, bokasi is a technique of composting through the fermentation process of organic material by adding effective microorganisms that shorten composting barrier. Thus, the empowerment of the Camplong to Villages community is required to improve better farming. Okay, so the study was carried out through interview with farmers, observation, and simple farming methods. And we use animal manure, effective microorganisms, burnt rye husk as the main component in the making of bokasi. And this is the ratio here. And the ingredient and the technical thing or the manure should be laid on the dry floor. And the common application of organic compound to the soil has traditionally been carried out through the incorporation of crop residues, compost, and organic residue. A promising aspect of the use of bokasi that was delivered to the community in Camplong too, including a high content of organic material and nutrients inside bokasi, and presents high porosity and high water holding capacity for the soil, which can increase the health of the soil in Camplong to Villages. Having shallow topsoil and rock formation, making it not suitable for cultivation that can affect the growth of the plant and its production. A mixture of recent knowledge and the traditional methods was used in the study. And so what we did, we applied bokasi in order to increase the soil, the health of the soil. The soil increased the organic material by providing nutrients and reducing material leaching. The small-scale agriculture product that has been produced by using this bokasi were eggplant, tomatoes, and peanuts. Men are not the main actors for supporting families' income, as women has also participated to grow some plants in their backyard and obtain benefit from using this bokashi. And the use of bokashi, we found that it is one of good alternative to improve soil fertility for soil with low quality, just like in Champlong to area. Bokashi contains living microorganisms, which when applied to soil, just like in Champlong soil, can support the growth of plant by supplying an essential nutrient. Therefore, this bokashi is essential to minimize the use of chemical pesticide and to support microorganism activity that can restore soil condition in Champlong to village. So, in conclusion, we found that the inclusion and implementation of small farming techniques in Champlong village led to beneficial results. While some of the people and its changes may not appear significantly, the small step will giant leap towards villager empowerment in Champlong to village. This is the references. And at the end, we would like to thank to the RPM Minister of Research, Technology and Higher Education, Brain, for funding the community service research, local farmer in Champlong to village, in Kupang Regency, and co-assistant student at Veterinary Medicine Faculty at University of Nusa Cendana, Kupang. Thank you for your attention. Hopefully this presentation will be beneficial for everyone. Thank you.
All right. Thank you for all the amazing presentation from our three presenters. So I would like to open our Q&A session. Wow, so many questions in the chat box. Okay, so I want to friendly remind you that you can send your question directly to the moderator or just to everyone in the chat box. So we will not miss it again. Okay, for the first question, it's for Mr. Desem. Okay, we have several questions here, but we will start from the first one. So the first one is, for the two misidentified isolation, did you continue with molecular identification? And what characteristic that make you convinced that those were Passerella mutosida when identified by traditional methods?
Okay, can you hear me?
Can you follow up?
Can you hear me?
We can barely hear you, sir.
Wait.
Okay.
Can you hear me?
All right, it's clearer now.
Okay, thank you for the question. And I would like to answer this question, but the first, what makes us confident about this isolate is Pastoral mutosida is there are two things in here. The first one is the identification method that conducted by Quant's algorithm, and I have to say here that this method is one of the manual for the identification of medical bacteria. That in this manual it says that there are two stages in identification the bacteria. The first stage is to identification its genus, and the second stage is for identification the species. In the first stage, we could say that these two isolates is the in the genus Pastorella. So the second stage, we using the so many biochemical characteristics, which most of them are in carbohydrates fermentation and utilization, and it is combined with the Berge's manual determinative of bacteriology. In two methods, we could say that isolate combination. We did it with the protein profile analysis, and it's not discussed here because we discussed it in another paper, and we could say that it is a quite high similarity with pastoral motorcida in other isolates.
Okay, but for the two misidentified isolate, did you continue with molecular identification?
By protein profile analysis, and we with the analysis with clustering, we could say that it's quite high similar.
Okay, I see. Maybe you can submit it again for the second ICAVESS. All right. So the second question here, I think we can jump to another presenter so that everyone get their first round of question first. So question for Mr. Didik. So would you think your method is applicable for rapid screening?
No, this is not designed for this. Just in case we could not isolate the live tachyzoid. Okay.
So and confirmation.
Yes. No confirmation. Alternative method. Alternative method when we can't isolate the tachyzoid, or we cannot growing in cell culture.
All right.
Okay. Yeah. We only have a dead tachyzoid.
All right. Yeah.
Because
if the limitation is on the tachyzoid, then you can use this method, right?
Yes. Because for pathogenicity of Toxoplasma gondii, only can do with bioassay in mouse or in rat.
Yeah. All right. So it's alternative, not for rapid screening. Okay. Yeah. Thank you, Mr. Didik. So moving on to the third question. It's for Ms. Cynthia. So the question is, have you done economical calculation for the end product of this bokesi? If I'm okay.
Hello, Ms. Andre, can you hear me?
Maybe you can volume up a little bit.
Okay, can you hear me?
Okay, it's fine now.
Okay, basically it's just this is like a community service, but we try to make another further research, just maybe an early research regarding the income that has been, income that can support the village's family, and there is differences between before and after the farmers using the bokasi from organic materials and or we call unorganic materials. They see the result, and from for the income results, they got the differences, especially for the economic background. For example, like in we did this in 2020, especially when pandemic is spread everywhere, so they have no choices. So agriculture is the main sector that boosts their income. Like in 2019, they have no idea, and they have only income from they got from livestock. You know the Bali cattle can support them, their family, 6 million from the livestock. But from this agriculture, they can add more. Yeah, like they make you see terong or lobak or something. They can get from 500 to 1 million for for the selling the product agriculture products.
Okay, so Bokasi also support the waste management, like the waste management for like food leftovers, right?
Yeah.
All right. Wow, that's cool. Okay, I think we covered everything, right? I think that is all from the question in the chat box. Thank you again for our amazing three presentator on the third subsession of this parallel session. Okay, we will now move on to the fourth subsession, which will be moderated by my co-moderator. Dr. Christiana, you may proceed.
Thank you, Andre. So we are going to have another three presentation. One from Bapak Aris Purwantoro. It's about toxoplasmosis molecular detection using polymerase chain reaction with 3 to 7 BP primer design based on Toxoplasma gondii B1 gene. And then on the second one, we'll have from Miss Oki Shafira Risti. So effects of biofertilizer application on growth rate and chlorophyll content of spinach in salinity stress condition. And also we're going to have the third presenter of the subsession from Bapak Damasus Rianto about the utilization of agricultural and livestock waste and the effect on new rice varieties yields on rain-fed rice field of Ponjong Gunung Kidul. For the co-host, the time is yours for the video. Thank you.
Thank you, moderator. We thank to the ICAVESS Conference Committee who invited us, and it is great honor to present our paper entitled Toxoplasmosis Molecular Detection using Polymerase Chain Reaction with 327 base pair primer based on Toxoplasma gondii B1 gene. The team consists of Aris Purwantoro, myself, Wayan Tunas Artama, Bambang Sumiarto, Adi Herustomo, Rini Widayanti, Agung Budianto, Widagdo Sri Nugroho, Elsa Herdiana, and Agustin Indrawati. Introduction. Toxoplasmosis is a zoonotic disease caused by an obligate intracellular protozoa Toxoplasma gondii. Toxoplasmosis, sometimes neglected, although it has medical and veterinary importance. Approximately 30% human population worldwide infected by this protozoa. As an archipelagic country with many rivers, lakes, and other water bodies, Indonesia should aware to toxoplasmosis since it is waterborne zoonosis. Most Indonesian farmers are rearing livestock as live deposit, which could be sold for instant cash. There are two routes of toxoplasmosis infection. First, ingesting undercooked or raw meat which containing viable tissue cysts. Second, ingesting food or water which contaminated with Toxoplasma gondii oocysts. Toxoplasmosis could be detected molecularly using polymerase chain reaction. PCR is simple, sensitive, reproducible method, and all clinical materials can be used as samples. Material and methods. 30 good meat samples were purchased from 15 kiosks throughout Kulon Progo Regency. The samples were extracted and amplified its DNA, then electrophoresis the PCR products. The PCR based on Toxoplasma gondii B1 gene was done conventionally with single-step method. The PCR also compared between commercial kit and 327 BISPER primer. Results and discussion. As can be seen on left figure, the PCR using commercial kit produced 406 BISPER band for positive samples of toxoplasmosis. On the right figure, the PCR using self-designed primer produced 327 BISPER band for positive samples of toxoplasmosis. This table is displaying association tests between commercial kit and self-designed primer of 327 BISPER. The calculation resulted 86.11% kappa value. It means that between commercial kit and 327 BISPER primer has an excellent agreement since it is higher than 70%. This study concluded that toxoplasmosis can be detected molecularly with polymerase chain reaction. The detection could be done using 327 BISPER primer, which designed based on Toxoplasma gondii B1 gene. Association test between commercial kit and 327 BISPER primer resulted 86.11% kappa value, which means excellent agreement for this method. Thank you very much.
Hello everyone, my name is Okti Sapira Risti as the first author and Dwi Umi Siswanti as the second author, and we are from Faculty of Biology, Universitas Gadjah Mada, Yogyakarta. It is an honor for us to have been given the opportunity to present the results of our research about effect of biofertilizer application on protein and chlorophyll content of spinach, Amaranthus tricolor L in saline stress condition in this event, ICAVESS 2021. So, spinach is one of the vegetable plant from Amarantaceae family that widely cultivated and consumed because of its high nutritional content. In its growth, environment is one of important factors that can affect its rate. An environment with high salinity can inhibit plant growth rate. To prevent inhibition of growth, plant needs to be given special treatment to help their growth, one of which is by applying fertilizer. The example is biofertilizer. Biofertilizer microorganisms that help carry out biological process so that it can help increase soil fertility and also produce antibiotic for plants, which is caused by the interaction between microorganisms in the biofertilizer and the rhizosphere. The aim of this research is to discover the effect of biofertilizer application on the growth rate and chlorophyll content of Amaranthus tricolor L in salinity stress condition. This research was held in August until October 2020 in Faculty of Biology Universitas Gadjah Mada Yogyakarta with the steps. First is planting of Amaranthus tricolor L plant in polybag with soil, husk, and manure as planting media. Second is treatment giving. So there were two treatments given in this research. The first is biofertilizer treatment that given to plant after plant are 2 weeks old with dose 0, 10, 20, and 30 liters per hectare. And then the second treatment was an ACL treatment that given to plant after on the 7th and 14th day after 2 weeks of seedling with concentration 0, 2,500, 5,000, 7,500, and 10,000 ppm. The next step is measurement of plant height and number of leaves. And then the last step is measurement of chlorophyll A, chlorophyll B, and total chlorophyll content on leaf. This is the results of our research. The top pictures show the rate of Amaranthus tricolor L plant height increasing on salinity stress condition on various salinity level. And the bottom picture show rate of Amaranthus tricolor L number of leaf increasing on salinity stress condition on various salinity levels. And this table show the chlorophyll content of Amaranthus tricolor L in this research. As it known that in this growth, there are several factors that can affect the rate of plant growth. Among them are natural factors such as sunlight, salinity, and nutrient availability, and also factors that are intentionally provided by human. For example, the application of fertilizer. Based on the measurement that has been done on plant height and number of leaves, the results shown that the highest growth rate of plant height and number of leaves was mostly in plants with 0 and 30 liters per hectare biofertilizer application. In this research, the plants that were given a treatment had plant height increase, which tended to be high, and there was an increase in several observations. It is because even though there was salinity stress in treatment plant, the stress given was not too high as indicated by plants that were able to continue to grow until the end of the observation. Apart from that, plants were also given biofertilizer treatment and also provided manure as basic fertilizer. So it can be one of the reasons why in this research, even though there was a salinity stress given through the application of an ACL, all plants can still grow well. The application of biofertilizer to plant in salinity stress condition can help increase the growth rate because the microorganism contained in the biofertilizer can cause colonization between this microorganism and the rhizosphere, which can help increase the soil fertility through increased nutrition for plant for growth. However, the dose given must be right. However, in this research, the results also show that the growth rate in terms of the increase in plant height and number of leaves in the B0 treatment was also high, even though the B0 treatment was a treatment with 0 liter per hectare biofertilizer. The occurrence of this error may be caused by the weight of other factors that influence the growth rate of spinach in that treatment. One of the most likely factors causing this treatment have a high growth rate in this research was the light factor. Because at the environment factor measurement, it is known that the B0 plant plot have the highest light intensity value compared to other biofertilizer treated plant plots. Soil and temperature are some of the important factors that influence the synthesis process of chlorophyll in plants. Based on the results shown in Table 1, it can be seen that the majority of plants treated with biofertilizer at a dose of 20 and 30 liters per hectare have a high chlorophyll content value. So it's known that the application of biofertilizer in dose between 20 to 30 liters per hectare in this research was able to help increase chlorophyll synthesis in leaves. But when viewed from the treatment of the amount of salinity, it seems that there is no value that indicates a pattern based on the level of salinity. This could possibly occur because the possibility of NaCl treatment given as a salinity stress did not really have an effect on the amount of chlorophyll content in spinach leaf plant observed in the research. Previously, in a research conducted by Halim et al. At 2019 regarding the tolerance test for spinach to salinity stress, the results show that the spinach still has tolerance at a salinity level of 8,000 ppm, which is also supported by provision of urea, SP36, and KCl fertilizers. In conclusion, biofertilizer application affect the growth rate and chlorophyll content in Amaranthus tricolor L that grow under salinity stress condition. In this research, the majority of land treated with biofertilizer application at a dose of 30 liters per hectare had a high growth rate and chlorophyll content in leaves compared to other plants in salinity 0 to 10,000 ppm. There are some references that I use for this paper. I think that's all from me. I'm sorry if there are any mistakes. Thank you so much and have
Okay, good morning to all of the participants on the seminar ICAVESS 2021. I would like to say thank you for the moderator that give me the time to present my research and the title of Utilization of Agriculture and Livestock Waste and the Effect of Rice Varieties Used on Ponjong Gunung Kidul. Well, let me introduce myself. My name is Damasus Rianto and I'm from IIT Yogyakarta. Okay, we move to introduction. As we know that the use of liquid organic fertilizer derived from chicken manure, live litter, and as supply of organic matter require an environmental friendly decomposer in the form of local microorganism. The use of organic matter decomposer is not only intended to isolate the decomposition process, but also to disinfect the compost material. Continue. For composting process can be isolated by adding an activator in the form of microorganism decomposition process. The decomposer activator can be made by breeding organism from the stomach, for example, from of ruminant animal. The improvement of organic fertilizer, which was not require a high cost, can be done by making of decomposer based on local microorganism. Okay, we go to the purpose of this study was to determine the application of various local microorganisms to the growth and yield of new rice variety in the rice field of Ponjong district, Gunung Kidul. For the material and method, the research site is on the Sidoarjo village, Ponjong district, Gunung Kidul Regency, and the research carried out from February to June 2017. For the tool research, use tractor, sprayer, analytical scale, tape measure, blender, plastic bag by volume 20 liters. And for the material, use Impari 19, Impari Sidenu, Impari 33, pesticide, fertilizer, urea, TSP, KCl, and fruit waste, vegetable waste, and glycida leaves. And for the treatment, we use 6 treatment and the research use completely randomized block design with 3 replication. For the treatment is following treatment M1, LMO vegetable waste, M2 LMO fruit waste, M3 LMO from banana weevil, M4 LMO from urine and co-manure, M5 LMO from glycide leaves, M6 is mixed from the 5 LMO ingredient in above, and rice variety we use in Pari 19, in Pari Sidenu, and in Pari 33. This one is the map of research site in Ponjong district, Gunung Kidul Regency. For the research and discussion, we can use table. We can see table 1, the highest, the total population microbes for the highest total microbes is reached by M6, a mixture of 5 microorganism ingredient, and for the nitrogen fixation bacteria, the highest is achieved by F5 from the local microorganism from glycide leaves, and the phosphate solubilizing bacteria, the highest one is achieved by M3 treatment that came from microorganism from banana leaves. For the macro element content, the highest for the phosphate available reached by M3 treatment, and the highest total highest is reached by M6 treatment. For the micro element is reached by M6 treatment. Actually, this one is complete element, although some element is still low content. For the table 4, so by the number of tiller affected application of various LMO, we can see for the highest reached by in pari 33 by treatment of 6. M6, and then we go to the table 5 is the number of field grain per panicle. The highest number is still in pari 33 in treatment M6. On table 6, for width of 1000 rice grain, the highest amount is still in pari 33, but the treatment came from the M4 and M6. This one is not significant. For the table 6, we can see for the week of harvest biomass, the highest result we can see in pari 33 in the treatment of M4 and M6. For the weight of harvest dry grain, the highest still in pari 33 is reached 8.92 and 8.79, and the lowest is Sidano in the treatment as M1 and M2. For the conclusion, the percent of micro and micronutrient content will change if the liquid of LMO fertilizer is made from the mix of organics, agriculture, and livestock waste. For the second, the highest content of macro element nitrogen is treated by liquid of LMO from gamma leaf, and the highest of phosphate is found in LMO with banana weevil waste, and from the highest key content came from liquid LMO, manure, and co-urine as much as 173.42 ppm. For the third, application of liquid LMO derived from mixed fermentation of agriculture and livestock waste give the total yield of whole grain per panicle width of 100,000 grain, and the yield of GKP higher in pari 33 variety than the application of LMO from other agricultural waste material. I think this one is all of my presentation. Thank you for your attention, and see you on other time.
Thank you so much. We have a variety of discussion today. So let's say, okay, for the Q&A session now, okay, we have one question for Pak Aris. So do you suggest that this 327 base pair could replace the commercial kit diagnostic test, or it is still recommended to use it as a complementary for the commercial diagnostic test?
Thank you, moderator. The 327 base pair primer could be used as a complementary for the commercial kit, because if we ordered the commercial kit, it takes more than 3 months arriving in Indonesia. But since this self-designed primer is cheaper, faster, and we use a mini thermocycler, which could be operated by battery. So it's possible to do a PCR in remote areas. Thank you.
All right. So, so with the commercial kit, is it a nested PCR or is it?
It's also a direct PCR.
Okay, so is it also based on B1 gene?
It's also based on B1 gene Toxoplasma gondii.
Okay, just wondering how many samples have you tried with this 3 to 7 compared to the commercial?
Actually, as a part of my doctoral dissertation, is more than 275 samples.
Okay, that's amazing. Thank you. Okay. All right, I'll have a look on the chat room. Okay, so we have a question for Mr. Damasus. What are the usage percentage for all ingredient in M6 treatment? Is it still achieve its maximum results if there's 1 or 2 ingredients missing?
I think you heard my sound. You heard my sound?
Sorry. Can you try again?
You heard my sound?
Yeah. Now it's clear.
I think in treatment 6, all of ingredient in the treatment 1 and 5 is collected and composed to ingredient 6. That one is treatment 6, and I think it's that one is already.
Sorry, your voice is interrupted.
That one is already optimum reach for the treatment 6. Hello?
Yeah. Okay. So it's saying that that the ingredient for M6 treatment is maximum. Is that what you're saying?
Yes, I think that one is correct.
Okay. Thank you. So we have another question for Miss Oki. I think I saw one for you. Just wondering why the highest concentration of biofertilizer was not giving the best result of spinach growth. Please, Miss Oki.
Firstly, I want to say thank you to Universitas Gadjah Mada for the RCA grant, so we are able to do this research. Thank you for the question. Actually, in my research, the highest dose of biofertilizer gives the best result on plant growth. But I think the dose given in this research was not the best for the plant for spinach. That is the highest is in research is 30 liter per hectare. But I think it's not the best, and I think we should do another research to find out what is the best dose of biofertilizer to give to spinach so it can help the growth rate. I think that's all.
All right, thank you. So good luck with your biofertilizer experiment for the next one. Okay, I think we have all questions for this subsession. So we're moving to the last subsession. So I'm going to hand it over to my colleague, Andre. Please, Andre.
Thank you, Dr. Christiana. Okay, so answering our last subsession of this parallel session. Okay, so it's been a long 2 day for ICAVESS. So hopefully we will wrap up this parallel session in a fruitful discussion. Okay, I would like to ask Ms. Susanti and Mr. Purwantoro to ready to turn on their camera. Okay, thank you, Miss and Dr. Haris. Okay, all right. Okay, so we will start the video presentation. So Please, co-host, you may start the video presentation.
Good afternoon, everyone. My name is Lily Susanti. I am from Environmental Science Faculty of University of Indonesia. Today, I would like to present my paper, Sustainability and Feasibility Assessments of Nanobubble Aeration Technology in Environment, Economic, Social Aspects of Penaos Faname Shrimp Farming. Penaos Faname shrimp farming in Indonesia is challenged by several issues in environment, economic, and social aspects. The study aims to assess the benefits of novel nanobubble aeration systems for ecosystem, business, and community. The environmental impacts of shrimp farming include development and operational impacts. The development impact is the conversion process from coastal land to shrimp ponds, which modify the coastal habitats due to infrastructure development. According to Pais Osuna in his 2001 journal, the operational impact is the release of nutrients or shrimp feed that is not consumed by the shrimps and become waste. It poses challenges to the sustainability of shrimp farming due to high inorganic nitrogen content such as ammonium and nitrate, which are toxic to shrimp species and can cause eutrophication in waters. For the economical aspects, Penaos Faname shrimp farming is known to be costly and usually capital intensive, as well as demanding considerable investment with an extended payback period. According to Brumat in 2007, shrimp farming should opt and invest on efficient technology that can lead to short-term economic returns. Therefore, the shrimp farming technology should yield high net present value within a short period. Moving on to social aspects. According to Diedrich in 2019, within Asia shrimp farming community, adoption of technologies, standards, and practices by small and medium-scale farmers has recently become popular and viewed as one of the solutions to increase income. Let's move on to materials and methods. The subject of the study was Penaos Faname postlarva 10, observed in nanobubble treatment and control. The nanobubble treatment used DO level 16 and 22 mg per liter, and we observed the total ammonia nitrogen over certain period. In this study, we used merged ammonium test kit, and the test was conducted at 8 and 24 hours. In terms of economic aspect, we focus on two variables, which are net present value and payback period. For the social aspect, we interviewed 36 people that are working in the hatchery. We tried to analyze the correlation between gender, age, and educational background to their preference in using the technology. As the result, for the environmental aspect, our study found that the use of nanobubble technology could reduce the total ammonia nitrogen by 9%. As for the economical aspect, we estimated that the net present value will be around 64 million, with the payback period of less than 2 years. For the social aspect, the Chi-square statistical analysis show that gender, age, and education has no correlation to whether or not the respondents prefer to use the technology. To conclude, the study has provided empirical evidence on how the nanobubble technology can impact the environment, economic, and social aspects. The result shows that nanobubble is feasible to be implemented because it can benefit these aspects by reducing TEN, yielding NPV, and gaining trust from the shrimp farmers. That's the end of my presentation, and thank you for your attention.
Thank you, moderator. We thank to the ICAVESS Conference Committee who invited us, and it is great honor to present our paper entitled Utilizing Livestock Manure by Anaerobic Fermentation to Produce Organic Fertilizer for Cultivating Low Glycemic Rice. The team consists of Aris Purwantoro, myself, Ariana, Trini Susmiati, Eko Wismawinarto, and Trisakti Haryadi. Introduction. Indonesia is a tropical country which has an ideal climate for rearing livestock. The livestock has potential to gain extra income and employment. Livestock species sometimes abandoned and left untreated, thus polluted surrounding environment. On the other hand, almost 2/3 human worldwide consume rice for energy source. Rice become popular diet due to cheap, fast, and easy preparation. Also could be combined into various cuisine. Furthermore, consuming rice with high glycemic index for prolonged time might lead to diabetes mellitus. Indonesia has position as 7th rank of diabetes mellitus patient in the world, with prevalence more than 0.2% per year. Materials and methods. Anaerobic digester was constructed of highly density polyethylene tank with 5,100 liter volume, which connected two polyvinyl chloride pipes in opposite direction. The 4-inch pipes serve as substrate inlet and fertilizer outlet. The digester anaerobically ferments feces of 3 cattle daily to produce organic fertilizer, which collected in 8 plastic tube of 200 liter volume. The fertilizer was used to cultivate Ciherang padi, which has low glycemic index rice of 54. results and discussion. The apparatus could digest abandoned feces to produce fertilizer and biogas. The retention time was 14 days with water dilution 2 times. Adding co-substrate such as sawdust resulted higher biogas yield. Human diabetes mellitus is a metabolic disorder of glucose distribution with low insulin level and higher blood sugar. The consumption of higher glycemic index diet will result higher blood glucose level. Controlling blood glucose level could be done alternatively by consuming low glycemic index food, such as ciherang rice with only 54 glycemic index. The study concluded that abandoned livestock feces could be fermented anaerobically to produce organic fertilizer. The fertilizer could be used to cultivate paddy, which has low glycemic index rice. The low glycemic index rice is suitable for human diabetes mellitus patient diet. Thank you very much.
Okay. Okay, thank you for the video presentation. Now we will, now we are going to start our Q&A session. We have 2 questions here. The first question is for Ms. Lily. So the first question is, although the acceptance of the farmer and it is proved to help them, however, do you think the nanobubble is applicable anywhere, like a larger scale?
For the question, Sir Andre, I think nanobubble has been introduced to shrimp faname in Indonesia and will help not only for the small scale farmers but also the medium and large scales. I also doing research for P. faname cultivation in shrimp ponds in Probolinggo and also in Anyar. Nanobubble technology can improve the water quality.
So it is
especially in dissolved oxygen level, and can reduce the oxygen consumption of shrimp until 66%.
Oh, that's good. So it's I think it's applicable in a larger scale, right?
Yes, sir.
Wow, that's great. I mean, if you want to produce a large scale, maybe you can. I think it's a large market in shrimp, so better be quick. Okay. For the next question for Mr. Aris Purwantoro. So the question is, okay, so there is 2 questions here. So the first question is, what's the key method or ingredient used for this manner to produce organic fertilizer for low glycemic rice? Maybe you can explain it in short. And the other question, it might relate to the first question. Could you explain like the paddy plant itself, like how different processing of the fertilizer will change its glycemic index?
Yeah, thank you. Can you hear my voice?
It's clear, sir.
Thank you. For the first question, the manure of 3 cattle just flow through directly to the biogas digester tank. There is no other treatments before entering the tank, the digester tank. And the second one, the paddy field already treated with this organic fertilizer for 4 times. So this is the 5 times application of organic fertilizer, and the product of rice increased significantly, more than 30%. And yeah, and compared to the non compared to the rest without organic fertilizer, the rice just sell to the consumers around 11,000 per kilogram. But the organic rice is higher, around 18,000 per kilogram, with better package and sell it through supermarket. The prices are more higher. Thank you.
Oh wow, I think it's a good research because you combine both the healthy side and the production side. So hopefully for the tour presentation here, this presentation can be met the market need, and maybe can help our society, especially our farmers. Okay, thank you, Mr. Purwantoro and Ms.
You're welcome.
Lily. Okay, I think we have no more questions in the chat box. Okay, I think we are in the end of our parallel session. So before we sum up this parallel session, I would like to say it is my great honor to moderate this parallel session. It is I immensely thank the OC of ICAVESS to let me moderate this parallel session, and I hope we had a fruitful discussion just now. I saw so many questions, and it was a long discussion, and I hope this diverse presentation from plants, animals, and diverse background, I hope it can make a sustainable development, just like this event purpose at the first. Okay, so I think I will, I think Dr. Christiana can take it from here. On to you, Doctor.
All right, thank you so much, Andre, for leading the session as well. So I'd like to thank you everyone for this great opportunity to share your expertise today. And I wish this very moment will initiate a new collaboration as well. So before we end the session, I'd like to ask for your permission to capture the moment. Therefore, could you please turn on your camera? Do we have everyone here? Okay. Do we have everyone? Arpendi? Aryo? In 3, maybe we can count, or not yet?
Oh, Mr. Sugiono is driving now.
Okay. Oh, sorry for
Stay
that.
safe, Miss Sir.
Okay. In 3, maybe? It's 1, 2, 3. Cheese. We good?
I think for the second page because okay. Again.
Okay. For the second page, 1, 2, 3. Cheese. All right, we got everyone. Yep. Thank you. So I also would like to remind you that there will be a dance performance, Mangasuti, and announcement of the best presenter. So don't miss out. Who knows, it could be you. So also we would like to hear your opinion on the ICAVESS. So please fill out our feedback form. The link is provided in the chat box and in the WhatsApp group. So again, thank you everyone. Also, thank you the Paralel team. Thank you Andre. Thank you Arpendi.
Thank you, Doc.
Aryo. Pleasure in making sure the session is going smoothly. Now you may leave the meeting and direct yourself back to the plenary room. Same link as this morning, and I'll see you there soon. Stay safe and stay healthy, everyone. Bye.
Thank you.
Thanks, everyone. Hope to see you offline.